Pode
Já
ser
está
isso, Marco?
gravando, já pode ir,
Can we start?
Yes, we can.
Perfect. So, primeiramente, eu gostaria de agradecer a todos que estão aí para a gente transformar esse momento desse webinário internacional um pouco mais acolhedor para o nosso convidado. Então, a gente vai falar em inglês. Mas vocês fiquem à vontade para vocês também poderem, aqueles que não estiverem confortáveis em se comunicar no idioma inglês, podem, por favor, utilizar português que o professor Jean vai nos auxiliar. So, it's a great pleasure to guest our Professor Olaf Sporns from Indiana University. It's an honor for us at Unifesp. He's one of the greatest names in the area of brain networks. It's pretty important for all the community of Unifesp to have you here today to talk about the new advances of this area. And I would like also to say thank you for Professor Jean Faber, who is in charge of this webinar. He was the one that invited Professor Olaf Sporns, and in the honor of the Graduate Studies and Research Office of the Unifesp, I would like to thank you both, Professor Olaf Sporns and Jean Faber, and let's start our webinar today. And we also have Professor Marimelia here with us. She's also a collaborator with the Professor Sporns, and she will be helping us to moderate this section. So, in order to introduce Professor Jean Faber, who is a member of the CapSprint team. So, Professor Jean is graduate in physics at the Federal University of Juiz de Fora, Brazil, and he received his master and PhD degrees in computational modeling in the field of quantum information and computation from the National Laboratory for Scientific Computing in Brazil. He then starts his studies in neuroscience as a postdoctoral fellow at the International Institute of Neuroscience in Natal, investigating the neuroelectrophysiological patterns during wake sleep cycles. He also did a postdoc at the Commissariat à l'énergie atomique et aux énergies alternatives in a project of a brain-computer interfaces. Currently, Professor is adjunct professor at the Federal University of São Paulo in the Department of Neurology and Neurosurgery, discipline of neuroscience. He is also interested in the areas of neuronal signal analysis, neural connectivity, and brain-computer interface. So thank you so much, Professor Jean, and the floor is yours.
Thank you, Camilo. It's me who has to thank you, actually, for the whole support to organize this webinar. Thank you, Professor Marimelia, as well. So it's a huge pleasure being here, and especially to announce Professor Sporns. It's a great privilege for us. Let me just introduce him to say some words about his academic life. So he's undergraduate in biochemistry at the University of Tübingen in Germany. Professor Sporns did his PhD in neuroscience at Rockefeller University in New York, and then conducted a postdoctoral work at the Neuroscience Institute in New York and San Diego. And currently, he's a distinguished professor in the Department of Psychological and Brain Science at the Indiana University in Bloomington. Professor Sporns earned John Time on Guggenheim Memorial Fellowship in 2011 and elected a Fellow of the American Association for the Advancement of Science in 2013. And well, besides the huge list of papers, I think it's worth to say that he has published the books Networks of Brain and Discovering the Human Connectome, both published by Mid Press. So his main research area is theoretical and computational neuroscience, focused on brain connectivity and network, Connectomics, including patterns of anatomical projections and the role of connectivity in shaping brain dynamics and functions. So, Professor Sporns, thank you very much to agree with this invitation. It's a huge pleasure for us, a great privilege to hear you. So I think the word is with you. Thank you.
Okay. Well, thank you so much for that kind invitation. I hope you can hear me loud and clear, and it would have been nice to be there in person, but it's not possible these days. So I hope this is a good replacement. I want to give you an overview of some of the themes and topics in brain networks that we've been working on here for the last 20 years in brain and animal models, and in human and animal models. And I will be available for questions afterwards. Since I cannot see anybody on my screen, have no feedback, so it might be good to, unless it's a very quick clarification, to hold questions to the end. I will be available. I have made some time after the talk to talk more with you about the concepts here. So.
Yeah. I'm sorry to interrupt. Yeah, I forgot to explain it. Let's make I don't know 50 or 1 hour per speech, and then we can make 30 minutes, around 30 minutes of questions from students. And
great. So I'll launch right into it. Hopefully able to advance my slides. Here we go. Brief introduction at first about this new topic of network neuroscience and some of the basics. And then I'll go into human connectomics, comparative animal model connectomics, and then the part of the talk I really want to get to is about dynamics and functional connectivity. We have some new work in this area that we're very excited about. I want to tell you briefly about this. Of course, many of you on this call know that brain networks have become a really big topic, and I'd like to remind us that this was not always the case. In fact, brain networks is a fairly recent topic in neuroscience, maybe 15, 20 years ago, when the field finally started taking off. And here on this slide, I'm just showing you a few iconic representations of networks in C. elegans, in the mouse, in the macaque, monkey, and in humans that have appeared over the last several years in the literature. And I will cover various aspects of brain networks in this talk. Now, I think there's a reason why brain networks are becoming a more and more important topic, and that is because we have access in neuroscience to more and more rich, large data sets, big data as we call it, all the way from molecules and their interactions in cells to individuals in a population interacting in social networks. This big space in the middle here, broadly speaking, Connectomics, deals with connectivity within circuits and within brains, measured with different modalities, recording techniques and approaches. I will focus mainly in my talk, especially later in the talk, on human data and on fMRI, and I'll explain more about that when I get to it. But basically, the availability of connectivity data for the first time over the last 10, 15 years has driven the emergence of networks as a new paradigm, and network neuroscience is a new field. Another schematic representation of how I sort of see the field converging. On the one side, we have colleagues who are working on molecular networks. On the extreme other end, we have colleagues working in behavior and social networks. We are kind of in between with our anatomical and functional mappings that we make in circuits and brains. Nicely, we have a common framework, a common analytic framework that we can deploy to make sense of network data, especially the important aspects of communities and distances and communication patterns. I'll say more about this later in the talk. The nice thing is that this network analysis framework allows for a common language, a common mathematical a set of ideas that can be applied to molecular, neural, as well as behavioral data. And that is what unifies the field under the rubric of network neuroscience. Just a few words about human connectomics. I was interested in the brain ever since I was in high school, really. Back then, the only knowledge we had, we're talking in the 1970s and 80s, the only knowledge we had about the human brain came from post-mortem dissection. This is actually a photograph. That what you see is a photograph of a brain that's been, obviously post-mortem, that's been removed and has been dissected to show these long-distance, so-called white matter pathways that connect remote brain regions to each other. There's, of course, a big advantage in doing post-mortem studies. You really can't do very many individuals, and you can only do them post-mortem, so there's no way to really check and correlate anything that you find with their behavior or cognition. About 10 years ago, 15 years ago, a new technique appeared on the horizon, and that is non-invasive imaging of the human brain. What you see here is a computational rendering, a reconstruction of putative white matter pathways in the human brain in a living person. This technique is now widely available and allows us to image and reconstruct anatomy in a large number of individuals. This allows us to relate features of anatomy to individual variation in behavior and cognition. This has been a true game changer for us. The technique appeared somewhere around the mid-2000s, and it was at that time that the area of connectomics really took off. My first collaboration in this field has been with Patrick Hagmann, at the time in Lausanne in Switzerland. This is the first map, the first connection matrix that we built back in 2008, actually 2007. And this is a matrix of that is constructed as follows. Each row and column in this matrix corresponds to a piece of cerebral cortex, a piece of gray matter. Each of the entries in this matrix that you can see here records whether there's a connection present or absent between these two pieces of gray matter. And the coloring indicates how strong the connection is. So here you see the right cortical hemisphere, here the left cortical hemisphere, and these are the callosal connections traveling through the corpus callosum. You can see there's symmetry between the right and left hemispheres, as we would expect. It is these connectome maps, these kinds of lookup tables or wiring diagrams, that have become extremely popular since we first published this account back in 2008. At the time, there were no such maps available. Now there are literally thousands, tens of thousands of them in the human domain that are available worldwide from many data sets. As we analyzed this matrix that you just saw, this connectivity matrix with graph theory and network science tools, several features stood out for us, and all of these have been reported originally in 2008, but have since then been reproduced many times in many different cohorts. Number one, every cortical region has its own unique regional connectivity fingerprint, as we call it. Its unique set of inputs and outputs, which we believe defines, to a large degree, its functionality. Overall, the connections are exhibiting a so-called broad or exponential degree distribution. That is, there is many gray matter regions have very few connections, and there's a few gray matter regions that have a lot of connections that are rich in connectivity, if you wish. Also, there's high clustering. Neighboring pairs of areas tend to be connected to each other densely and strongly. And there's short path length, which means that we can skip from one part of the brain to the other in a relatively short number of hops. Short path length is thought to facilitate communication. There are also modules, communities that are formed from densely connected clusters of regions that are interlinked by critical parts of the infrastructure that connect modules to each other. We call them hubs. And these hubs are interconnected into a structural core. So not only are there hubs that interconnect modules, but the hubs themselves talk to each other and are connected into a structural core, or as we now call it more commonly, a rich club. So many of these features that we gleaned in this very early study on literally the first example of connectomes have held up across not only the human brain, but also animal models, as you'll see in a minute. I want to say a couple of words about the rich club because it's a really salient feature in the human brain and also in animal models. Here you see on the left a schematic layout of a very simple network with just a few nodes that are shown schematically. And there are 4 modules here that are somewhat more densely connected internally. Each of the little circles here is a node, is a brain region, let's say, and there's a pathway between some of them. These nodes colored in black are those hubs. These are crucial. They have many connections. And they're also crucial for linking communities to each other. You can sort of see if we lose that connection here that links these two communities, it's very hard to pass information from one to the other. If you want to make an analogy to the global transportation network, these are your connections, intercontinental connections between major airports that are connecting continents to each other. Here are the regional flights that link airports more locally. In the middle, what we see is a schematic where all I've done is I've added a few more connections that interlink the hubs more directly, and that facilitates greatly how they communicate with each other. Think of these hubs as collecting information locally from their own community and then sharing the information in a global workspace, as it were, which we call a rich club. The term comes from social sciences. Rich here does not mean money, it means rich in connections, many connections. These are the red nodes you see here. They have many connections attached. And they're forming a club because they're preferentially connected to each other, more so than expected by chance. We strongly feel that rich clubs are important features in nervous systems because they evidently have a potential role in integrating information from many different sources and allowing quick and efficient transfer of information between them. Working with Martin Mannenheul for the past decade or so, Martin and I set out to look for the rich club for the first time in 2011. We found it on the first try. This is from our very first study here. The critical index is that red line up here, which has a peak, indicating that the rich club has denser connections than expected by chance. So we're comparing density of connections among rich club nodes to a so-called null model or a randomized set of networks. The rich club nodes themselves are shown down below. It's a little confusing to look at this in sort of a ball and stick diagram. It doesn't lend itself very well to visualize human anatomy, but basically you can see there are nodes along the cortical midline and the cingulate cortex and the medial parietal cortex. In the insula, the front of the brain is to the top. So we found rich club organization, and we also hypothesized that if nodes in the brain want to communicate with each other, want to send signal from one part to another, network science predicts that those communication paths must pass through at least one rich club node and perhaps even a rich club edge most of the time. Even though the rich club is a relatively small number of nodes and connections in the brain, it soaks up a lot of the traffic. It's very important for communication, and it's a prediction from network science, from the way we can lay communication paths through such a network. We also hypothesized that rich club damage, if a node were to be damaged by a traumatic brain injury, by a disease process, pathophysiology, what have you, that will have a disproportional effect on network integrity and efficiency. So the network will really be damaged much more if the damage occurs on a rich club node than in a more peripheral node. And there has been evidence over the past several years that indeed some disease conditions in humans do attack high degree rich club nodes preferentially, and perhaps therefore are particularly disruptive. That was all I wanted to say about human connectomics. There's a lot more to be talked about, obviously, but I want to be quick and efficient here and get through a lot of material quickly so I get to my last important new part on functional connectivity. So human connectomics is operating largely through non-invasive imaging of the kind I've briefly illustrated earlier. However, it is not the only approach to connectomics, and in fact, predating non-invasive imaging has been classic studies carried out in model organisms and animals, usually with the tools of histology, tracing tracts and white matter and gray matter connections after placing injections in the brain, and then looking where the dye or the label is traveling and reconstructing connectivity that way. In fact, there's a large database. There was a large database already almost 30 years ago that collected information about connectivity in the macaque brain, specifically the macaque visual cortex. Maybe some of you have seen this diagram. It's been sort of in every textbook since it first came out, and usually people point to it and say, "Look how complicated it is, and look how many connections there are." And that's true. That's a good perspective. But it is not a random network. There is order in this diagram on the left, despite the fact that it's hard to discern when you just look at it in an image like this. Here's the same network shown as a matrix. This is the exact same data set, actually, I've worked for many years on the macaque cortex with my late colleague Olaf. And we set out almost 20 years ago now to try to understand the topology of this very simple connectivity pattern. This is much simpler than what I showed you for the human. But the advantage here is this is histology, right? This is real fibers that have been traced and reconstructed. In humans, we can't do that for obvious reasons. And so the technique in humans is much more indirect. This is a much more direct picture, much more like what we call ground truth in the field sometimes. So this matrix here gave us a lot of opportunity to look at hubs, communities, even rich club organization, and we've done so over the years. With one of my undergraduate students, Logan Harriger, 10 years ago, we set out to revisit rich club organization in the macaque after we had discovered it in humans, because we wanted to be sure it wasn't an artifact of human imaging. It was not something that was just present in imaging data and nowhere else. We found rich club organization in macaque as well. If you're familiar with macaque anatomy labels, these are prefrontal regions, cingulate regions, orbitofrontal regions, and temporal regions, shown here in this diagram on the right. The red patches on this inflated macaque surface, brain surface, indicate the locations of these rich club nodes or rich club regions. What you can see is they are quite well separated. There's parts of temporal cortex, parts of frontal and orbitofrontal cortex, there's parts of cingulate cortex, all of them densely connected amongst each other, highly connected to the rest of the brain. And that is an expensive feature of the cortical architecture. I say expensive because the wires that you have to lay from these different regions of, connecting all these different regions to each other have to take up volume, space, energy, metabolism. And that's an expensive feature of the way the brain is wired. I'll come back to this idea of expense in just a few minutes, because it runs through the whole discussion actually. So we were very pleased to find the rich club in the macaque monkey. Subsequent to that, I worked with colleagues in Taiwan on Drosophila Connectomics for a while. We also find the rich club organization, modular organization in the Drosophila brain. We are on the cusp right now of getting a lot more highly resolved Drosophila brain data from laboratories in the US and overseas. So we will probably redo this analysis again at much finer grain of resolution pretty soon. Skipping to rodents, the mouse connectome was published, or a draft of it, now about 6 years ago by the Allen Institute, and was subsequently analyzed by us and others for graph theoretic network features. Once again, presence of modules and rich club organization in the rodent brain is evident, and is involving parts of orbitofrontal, cingulate cortex, parts of the striatum, parts of the part of the subcortex as well, all of which have analogs in non-human primates and humans. Very quickly, in my own more recent work with Larry Swanson at USC, in fact, ongoing as we speak, we visited the rat brain, various subdivisions over the years, and published extensive analyses on cerebral cortex, basal ganglia, hypothalamus, and thalamus so far. We're currently working on the entire forebrain. This matrix here that is showing the bilateral cerebral cortex of the rat, 77 times 2, 154 regions altogether, was constructed by examining the existing literature on rat connectivity, existing literature from track tracing and histology studies, work that's been published in some cases decades ago. Larry and Joel Hahn went through the literature literally by hand and created a database that for each entry in this matrix here records whether there's a report that actually reports on connectivity being present between the two regions. The labeling is here on the side. If you're familiar with rat nomenclature, there's visual and auditory cortex over here, orbitofrontal regions. I see a few orbitofrontal regions down here. I see, where's the claustrum, one of my favorite regions. There it is, CLA. So 77 regions times 2, 154. Each of the entries here corresponds to published literature in histology about whether a connection is present or absent, and also how strong the connection is. This is a directed matrix, so the connections can go from A to B, but not from B to A. There's no symmetry here. This is actually not a symmetric matrix, but an asymmetric matrix. And there's evidence of connection strength as well. We have set out with Larry in a series of papers now to analyze different rat subdivisions of the brain. Why the rat? There's a lot of histology and literature already, and it's one of the model organisms that people use for behavioral studies in laboratories throughout the world. So there's known homologies from many rat regions to non-human primate and primate, non-human primate and human regions as well. So it's a model organism for that reason. One final point about the rat I want to make is this a methodological point. I've mentioned for a few occasions already the importance of communities. Just to review once again, a community in a network, broadly speaking, is a dense cluster, a cluster of nodes that is more densely connected internally than expected on the basis of a random null model. There's a comparison involved to a null distribution that says, how surprised am I to see these regions densely connected with each other? That's how we detect modules in a data-driven way. Modules, however, don't just exist at one level of scale. Now, in a network such as this one here, which is the rat end brain, the conjunction of the cortex and the basal ganglia, we can actually look for affinities or co-classifications between pairs of regions at multiple levels of scale. And that gives us the following matrix that you see here, shown in the same ordering scheme, which is a so-called co-classification matrix that says for each node pair, again, rows and columns here are regions of the brain or nodes. For each of the node pairs, how strongly affiliated are they with each other? How strongly coupled are they in a community sense? If it's white, if it's very bright looking here, these are regions that are almost always found together in communities, no matter what scale we look at. If there's nothing, if it's black, that means those node pairs are never found together. They really are very, very remote from each other. It's sort of like a distance matrix that tells us something about the separation of pairs of regions in the brain with respect to community structure or modules. This is a very fruitful approach, has been very fruitful for us in the rat brain, and we've extended it now to the whole forebrain, including hypothalamus and thalamus, with some interesting results forthcoming shortly. It's also a technique that is applicable to any network that you can possibly think of. This could be done on a transcriptome network. This could be done on a social network. In fact, the colleagues I've worked with on this are in computer science, and they work with social networks predominantly. One of the nice things about network science approaches is that they really apply to networks no matter where they come from. And that is one of the advantages of the framework, I think, over others. So we've been very, very eager to exploit this graded scheme of co-classification in both model organisms as well as in human data. And it's a methodological advance that I find very fruitful. I want to say just a couple of words about constructing networks from circuits or from individual neurons. So far, I've talked about whole brain, whether it's non-invasive imaging or histology, it's always whole brain data I've been talking about. It's actually quite a laborious process, and we happen to have, Gene, I don't know if you mentioned in your introduction, but we happen to have a PhD student from your lab with us here in Bloomington, unfortunately in quarantine. And we've been working very, very closely the last few months, last few weeks on trying to find better ways of extracting network representations from essentially spike trains, or let's say more broadly, point process data where we have zeros and ones in a row. I've worked on this with my colleague here at IU, John Beggs, over the last decade or so. And one of the techniques that John likes to use and that we've been looking at with him from the point of view of network analysis is transfer entropy, which is a way of estimating the impact of a neuron J's past state on the future prediction of another neuron I, in addition to what that neuron I's future state is predicted by from its own time series. Does this neuron make a contribution to predicting the future of neuron I better than just neuron I on its own? Sort of the gist of it. And in a nutshell, that tells us something about potential causal influence. That's the interpretation of transfer entropy quite often. Now, to extract transfer entropy from real spike trains or point process data is very hard to do. What we are looking at here is the enormous amount of false positives that have to be ejected, retaining only a small number of couplings that are strong enough to survive rigorous jittering and shuffling and other null model operations. When we do that, we get, this is work with Sunny Nigam, one of John's PhD students at the time. We can, John is recording from slices in a dish. The slices have been placed on a multi-electrode array, 512 electrodes, and he can record with that setup from several hundred neurons at a time. Each of the points in this diagram here is a neuron shown in space as it's laying down on the cortical, on the slice surface. And the connections, of course, are transfer entropy connections that have been reconstructed from those neuron spike trains. And the one point I want to make, this is a fairly elaborate paper otherwise, is that when we remove connections by strength, so we first remove the weakest connections, then the next weakest, then the next weakest, we actually continually see a connected network, and the density of connections that we observe is much stronger than expected by chance. That's the diagram here on the right, and that is the definition of a rich club. So much to my own surprise, but frankly, I did not expect to find rich club organization at the circuit level. We did find it, and so that is the third, fourth, fifth separate identification of rich club organization now in humans first, then in macaque, then in mouse and rat, and finally in circuits as well. It seems to be a very universal feature of how circuits and brains are organized. Coming to these common features and wrapping up this part of the talk, I mentioned several of them, especially modules, hubs, and cores throughout the last few minutes. Just to reiterate, wherever we look, whether it's C. elegans, which is a single, a very small nervous system composed of about 300 neurons, whether we look at circuits, whether we look at mammalian brains at large scales, we always find that there are broad degree distributions. There are many nodes that have relatively few connections, but there's a few nodes that have many, many more. Those are our putative hub regions. We're finding that there's unique connection profiles, whether it's single neurons in C. elegans or brain regions in the human. We're finding small world properties such as high clustering and short path length. Always find modules which are densely internally connected but more weakly connected between. And we're finding that hub regions are integrating information apparently into a core or rich club organization. This is true no matter what the scale we're looking at, no matter what the animal species is. It seems to be a universal set of features. Why is there this universality? Why don't brains look totally different one from the other with respect to their topological properties? One of the reasons why that is so is because a lot of what brains look like, especially the anatomical brains we've looked at so far in this talk, a lot of it depends on issues of wiring cost and network efficiency. Neurons and brain regions occupy space. They are arranged in a given geometry. They have physical distances from each other, measured in micrometers, millimeters, or centimeters. And the connections that they make with each other anatomically require an expense of volume, a certain length, and therefore delays in transmitting information, and also energy, because the wires, the axons, are living things. They're not made out of copper or whatever. They're actually little cytoplasmic conduits that are built from lipids and proteins that turn over every few hours. So there's a lot of energy consumption involved in making connectivity in the brain. And all that energy and volume is limited. The brain cannot expend more energy, more ATP, basically, than it actually has available to itself. Unlike in the real world, there's no way to run a budget deficit. The budget deficit will kill you. So you have to be able to allocate energy efficiently, and you can only afford the wiring that is compatible with the energy budget, with the amount of volume and the amount of energy that you have available to build the brain in the first place. That places huge constraints on how brains are organized. Many potential connectivity patterns we can dream up in our own minds or in computational models simply are not feasible because they would take up too much volume, too much energy, they can't be built. That is why I think that's what's driving, at least one of the things that's driving these common properties that we find across many, many different nervous systems. And we have, together with my colleague Ed Bullmore, have written up many years ago now, we should probably do revisit this at some point, sort of a simple, I wouldn't call it a theory, but a simple framework for how to express this. The actual topology of nervous systems is complex. It has a mixture of local modularity and long-distance connections, such as Rich Club, which are expensive. If all that we wanted was to make the brain as cheap as possible, the brain would look like this. All connections would be very short. You can see right away that it would be impossible to pass information from the back to the front. So that's great from a point of view of keeping wiring short, but it's really bad for performance. Strangely enough, and we can discuss it later, in a random network actually is the most efficient in moving information around. You have the shortest path length possible in a random network. However, it's also the most expensive to wire. Most of the wires here are really long. The brain would have to be a kilometer across, it turns out. If we were to wire the brain randomly, we cannot do it. So what's actually happening in the middle, the actual brain we have, is a compromise or a trade-off, largely involving lattice-like topology, local features of connectivity, local densities, and then also an admixture of long-distance connections that resemble random topology. So it's a trade-off. It's basically a compromise between these two opposing tendencies. That's what we feel is it's a good sort of, first-order approximation of what might be going on and why brains look alike, whether they are in C. elegans or in humans. Last part of the talk is the part I want to spend a little bit more time on and go into a little bit more detail because I'm excited about it. I've been working on it. Some of the things I'm going to show you I've been working on myself actually for the last several months, and I want to see what you think. So far I've talked only about anatomy. When I've been referring to Connectomics, up to this point, I've been talking about wiring, axonal connections, connections between neurons, things that are physical and real, and sparse for that matter, because the brain is fairly sparsely wired. But now, what about going from a skeleton, an anatomical skeleton shown here on the left, our famous connectome picture from the human brain, lateral view of the human brain. How about what's going on when the brain becomes active? I really like this little video. I've shown it many, many years now, almost in every one of my talks. It's actually taken of a person in an MRI scanner who is not doing anything in particular. This person is just lying flat, keeping their eyes open for a few minutes, and the scanner is listening in on what the brain is doing. This is the brain sort of talking to itself. This is now called, in the field of human MRI, it's called resting state. We can discuss later why that's a misnomer. It's not, the brain is not resting. As you can see, it's wildly active, even though the person is resting. And it's not a state because it doesn't stand still. It's actually dynamic. It's moving all the time. Each of the orange patches here is an MRI activation. That's a bunch of, large number of neurons becoming active and firing. Each of the blue regions here are parts of the brain that are somewhat inactive at that point in time. Looking at a movie like this, it looks really chaotic, right? I mean, it looks like it's just a swirling mess of activity. And it turns out there's a lot of structure in this, despite the fact that resting state is a is a state that doesn't require any instructions to the individual. There's no specific task being performed by this person in the scanner. So the brain is not challenged in any particular way. There is structure in this activity. And what has occupied us in the lab for many years is sort of this question mark in the middle. How do we get from the anatomy on the left, which is sparse, fairly static, doesn't change that much, at least not on these time scales, and something which is as dynamic, almost like the surface of the ocean, you know, here on the right. How do we get from left to the right? What's the mechanism? What's involved? So to unpack that, I'll tell you give you a very brief 2-minute primer on functional connectivity and what people understand by it in fMRI research. So we take a movie like this in a person that's in a scanner. We literally take frame by frame, we take pictures of their brain activity. Effectively, think of this as a vector of activity across the cortical surface. We take many such frames or pictures and we string them up as a movie, as you've seen before. And we then cross-correlate the time courses. This is super simple to do, although a lot of expertise goes into the pre-processing of the data to make sure it is denoised and unbiased and properly aligned and put into an anatomical template and so forth. We spent a lot of time in the lab dealing with data pre-processing, especially in the fMRI arena. But once we've done that and we have those time courses, the ups and downs of the bold signal for each node of the brain, we do something very simple, a cross-correlation, Pearson correlation, very simple. And we get a correlation matrix that you see here shown with the correlation values mapped onto a color map. If we cluster this matrix properly, we see there's real structure to it in the sense that there's coherent blocks of regions that are highly coherent. That is, their time courses are very similar to each other, and they're forming what we now call functional systems. The nomenclature here is things like visual, somatomotor, saliency, ventral attention, dorsal attention, default mode, control. These are names that have been given to these systems by researchers over the years, in part because they have connotations with cognitive function, it turns out. And this is very reproducible. This is a single individual, I believe, from a study we published many, many years ago. Every one of us, everyone on this call, everyone in this webinar has a default mode system. This system here, which has a particular topography to it in the brain, is universal. All of us have it. And we can find it and see where it is by looking at resting state functional connectivity. This is the amazing breakthrough that happened about 10, 15 years ago when people realized you could actually map the brain using activity where there's no instruction, there's no task, nothing at all. Down here, an early set 2 studies published back to back by 2 different research groups, one in Boston, one in St. Louis, using very different subject cohorts, mapping these resting state networks that you see here as blocks in a matrix, mapping them back onto the brain surface. The pink or salmon colored assembly here is the default mode that has parts of the medial parietal cortex, medial orbitofrontal cortex, temporal cortex, etc. Almost the exact same arrangement shows up in this diagram on the right, except here it's shown in red. Again, two papers back to back, same year, different journals, different groups of people that were imaged, different processing strategies. Same systems popped up in both of these studies. This was a real breakthrough and showed us a way to map out the human brain we've never known before. It's now called functional connectomics. So this is the gist of functional connectivity, at least resting state. And in our own lab, we were interested early on to model this. So how do we actually can we make a computational model that reproduces some of these features of resting state functional connectivity? The way we do that is we take an anatomical matrix, which is a coupling matrix, essentially, that couples different nodes to each other with a certain coefficient. And we then place at each node a set of differential equations that describe how that node is behaving through time, excitation, inhibition, type dynamics. oscillatory dynamics quite often. So essentially, we're looking at a system of coupled oscillators, and with some trickery and some specific biophysical mumbo jumbo thrown in, and some fMRI, synthetic fMRI equations on top, we can basically generate synthetic fMRI signals from such a system. It's nothing more, nothing less than a system of coupled differential equations that gets integrated numerically. And we get time courses from that. We can then build functional connectivity matrices and compare them to empirical matrices. Here in this case, the model preceded the data by 5 years. We had published the model in 2007, and 5 years later got access to monkey data that was comparable, and we're very pleased that we had a fairly high agreement between the two domains, despite the fact there was no model fitting. Later on, with my postdoc Genie, we built analytic models, actually models that do not require a full-scale biophysical simulation, but actually we can carry out in graph theory in MATLAB in a half a second. And we can predict functional connectivity. The right part of this fancy diamond-shaped figure is the prediction. The left part is the empirical. On the left part, you have real people, fMRI signals, cross-correlated. On the right, we have a prediction of the model based on those same people's anatomy. And you can see there's a very strong agreement between these two. So where does resting state come from? It's shaped by the anatomy. It is shaped by the underlying wiring diagram that we get with Connectomics, shown here at the top. And if we put dynamics on top of that, what we get is a resting state spontaneous activity. What we found early on in our computational studies is that this movie that you saw earlier, here's one of our own movies made from a simulation, actually is non-stationary in the sense that the statistics of which part of the brain is strongly driving the rest of the brain actually change a lot through time. And we call that hub dynamics. The status of a region as a hub is actually not fixed in time. It actually changes across time if we look at these dynamic assemblies. And other researchers since have developed this into many, many different directions. This is now called dynamic functional connectivity. And a lot has been written about the relation of this phenomenon observed in humans, but also in model organisms, by the way, and how it might relate to multi-stability and near criticality in human brain dynamics. One of the things that my former postdoc Makoto Fukushima found in looking at small data snippets taken from a movie that you just saw, and just looking at sort of windows of time, small windows of time, one of the things he found was that the modularity of the pattern that he saw was changing. Sometimes modularity was really low, sometimes it was really high. And with very elaborate techniques that he developed while he was in my lab for 3 years, we're still working together actually. One of the things that he found was these fluctuations in modularity. And he also found that there were recognizable patterns that the brain sort of switched into when modularity was high. Modularity means, once again, we're looking at communities of densely coupled, densely coherent sets of nodes. When those communities are very strong, very strongly expressed across time, when the so-called Q function or Q metric, which tells us how strongly modular the system is, when that is maximal, there's a recognizable topography in the human brain in the sense that we see disconnections between the default mode and control systems and the systems that are more involved in directed attention and multi-sensory information. So there's a recognizable topography that the brain snaps into when the modularity is high. Instead, when the modularity is low, the connectivity structure, functional connectivity structure is rather flat. And that was an observation that held across 2 different data sets involving each of them hundreds of individuals that have been imaged in various parts of the US, as you can see in this diagram. We've been on the sort of pursuing this angle for a number of years now, and I want to briefly mention last 10 minutes or so the work that we have just been conducting the last year or so. It's been an exciting year, I must say, for us in our lab and in the lab of Vic Betzel, whose lab is right next to us. We're working very, very, very closely together. Rick and my grad student Josh Faskowitz actually came up with an ingenious idea, which is the following. So far, I've told you about functional connectivity as the cross-correlation of node activations, essentially the ups and downs of the Bolt signal measured at a given region. And that was the Pearson correlation of the node time series. That was the functional connectivity definition we've used so far. And hundreds, maybe thousands of papers have been written so far, thousands of papers, literally, that have used this construct mostly in humans, but also in animal models. It's been an extremely popular way of looking at brain activity and connectivity for the last decade or so. Josh and Rick came up with the following idea. What if we started looking at not the nodes, but the edges as our primary construct of interest? Here's how this goes. So I'm going to show you two Bolt signals, two regional time courses here schematically. This is the ups and downs of the Bolt signal, right? For each pair of two regions, I and J. And in computing the functional connectivity, one definition of the Pearson correlation is it's the average of the product of the standard scores. So what we can do is we can multiply these two time series together. We get this construct here, which is now another time series that is between nodes I and J. So that's an edge time series. That's an edge because it's a connection I to J. Okay. It's not a node anymore, it's two nodes and how they talk to each other. That's the idea. The average of this time series here is equal to the functional connectivity. Let me go back for a second here. This is important because we're not inventing anything new. We're just suggesting now, the main suggestion is let's not do the averaging step, but instead let us keep all these time series and analyze them and look at the connectivity between edges. And you might say, what is that all about? And hopefully I can explain it to you in a minute. So each of the rows in this matrix here is an edge, is a pair of nodes talking to each other, and that's an imaging run containing about 900 frames or 900 time steps. So that's basically a raster of activity over time for, in this case, about 19,000 edges. And each of them corresponding to one pair of nodes. There's 200 nodes altogether. If we average this matrix in the y direction, we're getting a vector. If that vector is folded back into matrix form, that is equal to functional connectivity, what I've shown you earlier. This is the construct that people have been using in human MRI research, for a decade or so, with thousands of publications. Okay, that's all it is. And what we're proposing is let's not do the averaging step and let's not do this. Let's keep this construct and let's study it because there's interesting structure. Perhaps you can already see a little bit of it, even with the naked eye. There's some interesting features here that I'll come back to in a moment. We call these time series co-fluctuation time series because they tell us something about the fluctuations of the two nodes and how they operate with each other, whether they agree or disagree. We argued in our publications, one of them coming out soon in Nature Neuroscience, probably this month, another one currently in revision, that these time series tell us something potentially about communication processes in the brain. We can take each of these time series and we can correlate them. And that now gives us a much bigger matrix. This is regular functional connectivity between nodes, 200 by 200. This is a functional connectivity matrix between edges, 19,900 by 19,900. You can imagine this is pushing against the limits of what a regular, even a fairly powerful workstation can handle. Much larger matrix than we typically deal with. But it has information in it that we find very useful. And this is edge functional connectivity, or EFC. Turns out it has its own community structure. We can cluster this matrix. Now this is a clustered matrix of edges. We can map these edges back onto the cortical surface and then find that the edges overlap on nodes. And this is because when you cluster edges together, this is a little schematic diagram here from the work of one of my colleagues at IU, When you cluster edges together, these blue edges are belonging to one cluster, the green to another, the red is in a cluster of its own. They intersect with nodes in a naturally overlapping way. So nodes are now members of clusters of edges, of different clusters of edges. There's overlap, okay? This can be visualized on the cortical surface, the degree of overlap with an entropy measure to tell us how diverse the overlap pattern is for given sets of nodes across the brain. We have found this very informative, again, in our publications, which are both posted on bioRxiv. We dissect all this in great detail, and I encourage you to look up some of the detail. I'm not able to, I'm looking at my clock here, I'm not able to cover a lot of it in this talk, but this is just an illustration to show that there's different data sets from the Connectome project, the so-called Midnight Scan Club, which is a data set collected in St. Louis, and get another data set, and they have sort of similar profiles in terms of this normalized entropy. Many more findings contained in these two preprints down below. One of the things that we are excited about is that we find functional connectivity is modulated by sensory input. We have looked at this in movie data, where people actually are not just resting, but actually watching a video with their eyes open. And we're finding interesting patterns within and between individuals. And it does seem to have a stable, consistent signature also within individuals across different imaging sessions. So there is something in the pattern of this network that is individual specific, allows us to map individual traits back to brain connectivity. We are interpreting regular functional connectivity, node communities, the classic construct, as patterns of co-activation, while edge communities allow us to see patterns of co-fluctuation, perhaps communication between brain regions. Final paper I want to briefly review is by Farnas Esfalani and co-authors here. That's the one that's still in revision. I'm going to go back to this previous image of the edge by time matrix. You can perhaps see with a naked eye, there's some interesting features here, right? You see these vertical streaks running through this matrix. When we first saw those, the natural reaction of anybody in MRI research is, oops, we got an artifact. Because MRI, unfortunately, is a great technique, but it's also very sensitive to physiological interference, heart rate, breathing, head motion, involuntary head motion, things like that have a bad impact on signals quite often, have to be carefully expunged. So when we saw these streaks, our first fear was, oh, we've got artifacts in our data. It turns out not to be the case. If we actually compute a summary statistic for each of these time steps that informs us about the amplitude of these signals, each of these frames now taken to be one vector, this is what we get. This is basically a simple root mean square of the activations that you see here. And there are these very punctate intermittent epochs that we call informally events, when the amplitude of the signals is very large. These are the streaks that you see. And something is going on in the brain here, okay? This is a scan of several minutes. Intermittently, there are these epochs when the brain seems to be going haywire. There's a lot of amplitude in the signal, and these are intermittent. They don't happen very often, only every few minutes. Notice the time base. This is 900 seconds. What is 900 seconds? I can't do this. 15 minutes, right? Something like that. So it's a long period of time. We don't see very many of these, but we see them occasionally. And it turns out they have reproducible structure to them. These events, which are sort of bursty outbreaks of activity in the brain, they're unrelated to head motion or cardiac respiratory signals, as we've established. And they are present in all individuals that we've seen so far, and present in resting state, but also in movie data, interestingly enough. And these events have a particular signature to them. I'll skip right to the conclusion. If we actually take patterns of activity while these events occur. I'll go down straight to the bottom left panel in the interest of time. There's a consistent topography to it, involving activity in the default mode and control systems, and depression of activity everywhere else. And this is reminiscent of those high modularity epochs that Makoto and others have seen in their work during earlier years. So we're finding that events, these brief intermittent periods, have particular spatial signatures in the brain, are not just uniformly randomly distributed, but have particular patterns to them and recur. And we have since been digging into these events and what they mean, where they might come from, their relationship to behavior, their relationship in movies to content of the video signal itself, their spacing, their amplitudes, all sorts of attributes that they might have. And I found very interesting things, again, that are laid out in more detail in those publications below. Final, almost my last slide, just to put some of the ideas back together and into a common reference frame, the way I think about things, as it were. I've talked a lot in the earlier part of my talk about anatomical connectivity, structural connectivity, wiring patterns, sometimes weighted, sometimes directed, and multi-scale from circuits all the way to the whole brain. That's the anatomy. And to me, the anatomy is fundamental. I attribute that notion to my training as a biochemist. You know, when I was mentioning in the introduction, my undergraduate degree is actually in biochemistry. And what got me into biochemistry was my fascination with protein structure. And how amazingly detailed the picture of function was when you knew what the structure looked like. If you want to know what a protein does, where it is in the cell, what it interacts with, the first step to understanding that is to understand the backbone of the peptide chain, how it's folded, its surface features, etc., the structure of the protein. That's how I approach, rightly or wrongly, that's how I approach the brain. Anatomy is fundamental. This is our connectome down here, a static, fairly slowly changing wiring pattern that is sparse and connects different regions of the brain with each other. Within this network, there's a lot of activity percolating back and forth. I mean, the number of action potentials that is actually unfolding in our brains right now is in the trillions, okay? Every one of us on this call, our brains are furiously active. And when they become active, of course, signals are being passed, messages are being passed between neurons, and they travel along axonal pathways. So this is the skeleton. This is the router, the internet router network, if you wish, for all the traffic that unfolds on top of it, which is dynamic, fast changing, and involves signaling events. And then what happens is neurons and brain regions change their behavior. They turn on, they turn off, they correlate with each other. That is what we capture in functional connectivity. Those signatures of how different parts of the brain relate to each other over time, those statistical dependencies, those correlations, that is functional connectivity, and that's what we can measure. I want to point out that we can measure the anatomy, maybe not perfectly well yet, but we're getting there. We can also record with fMRI, for instance, functional connectivity, or with transfer entropy in circuits. We can measure how neurons are interacting with each other via spikes. We don't have very good tools yet to really get this middle level of the signaling itself. We don't see the signals themselves. We see only the wires, and we see the manifestations of the signals in time courses. This is hidden from us currently. It's a hidden domain of connectivity we do not see. And I feel that that is one of the great frontiers in our field, to understand how to actually measure this and how to model it. I've come through these various stages in my talk. Hopefully, I have no idea whether you're still with me. I'm looking at my screen, talking to myself in my office. This is I guess the new normal that we live in. But I hope you're still there. I don't know, but I'm hoping. And so we've gone through a bunch of different topics here in this talk, finishing with what I hope is I've been able to drive across to you some of the new ideas we're working on, potentially moving functional connectivity and brain networks from a node to a more edge-centric perspective. We can talk more about this in the discussion, and the importance of communication dynamics. in my ample spare time, I've also started a new journal at MIT Press, open access, online only, network neuroscience that is dedicated to all the topics that you've seen here. Perhaps if you have a moment, I invite you to browse the recent issues and the articles we published so far. With that, I do my acknowledgements and my thanks, and I'm now going to stop sharing. And I hope very much that I will still see you on the other side. If I lose connection, I will reconnect. I'm going to stop sharing, and hopefully, let's see, exit.
Everything is working well.
Okay, good. Oh, I'm glad. I have no idea. Okay, there we are. We're back in the. So I can see myself. I can see at least you. You know, this is good.
Yeah, perfect.
Okay, so that was my talk. Thank you very much once again. I'm very eager to hear your questions. I'm sure I've not explained everything perfectly, so there's probably many things we can dig into, and I'm happy to do that.
So thank you, Professor. You know, it was an amazing presentation. We already have some questions here. So let's start with them. Let me select one. Vinicius Carvalho asked you, what is the relationship with brain plasticity and topology? For instance, rich club regions more plastic and reshaped by learning, or the opposite?
Well, this is a really good question, and we don't know the answer to that. And in fact, it's in part of what we're trying to find out, working with you and with Priscilla, right? Looking at developmental patterns in cultured systems where we can measure them in a dish with electrophysiology and imaging. How does this connectivity that I've talked about, let's say the rich club, where does it come from, right? Is it there all the time? Probably not, because we know the brain starts out with many fewer neurons and connections. But then what drives its emergence? How does it change as learning occurs, plasticity? Plasticity is a difficult topic for us on the human imaging side because we can't measure it very well. We don't have access directly to the physiological strength of connections. We can only measure time courses of fMRI, and then we can measure anatomy, but we don't have a good sense of how strong the connections are. It's unfortunately a gap. But we can do that in model organisms and in culture. And so part of the things that Priscilla has been working on over the past several weeks and months is to look at time courses of topology, really. How does topology change as a system grows? Okay, as we observe it growing, as more connections appear, does that change the topological properties that I mentioned earlier? For instance, does it appear right away as a small world, or is that something which pops up later, or is it even more small world early on and then becomes less so? These are really open questions and exciting questions to be asking. And in the human domain, one thing we can do is we can look at people at different stages of development. So I have worked with Patrick on many years ago now on imaging from kids, from children that had gone to the hospital with a condition that had been imaged. And we were looking at how connectivity changes in children as early as 2 years old. And it turns out that the surprising result at the time was that a lot of the features that I mentioned to you, like modules, communities, even Rich Club, are present early on. They see, at least as early as we can look with these imaging techniques, let's say in infants, we seem to see some of that. And so it's not something that happens really late in development. It's something that happens pretty early. That's our current level of insight.
Yeah, maybe a good approach would be to look at, for example, amputee patients, because we know that they will go across a severe plasticity process, especially during rehabilitation. So maybe it would be interesting to see before and after the rehabilitation to try this plasticity, no?
That's a great idea, and I think people have tried to do that. Here's the difficult part of it, though. I presented non-invasive imaging as sort of this magic tool that allows us to see all these great things, and it is, at some level, it is really powerful. But as I also mentioned, it is susceptible to a lot of noise, artifact, and distortions and things that unfortunately make the signals that we see somewhat obscure. And so having really high quality data on multiple visits, on multiple, let's say pre-treatment, post-treatment, is really rare still. There are very few longitudinal studies that have really looked at people at different stages of development, the same people, not different people who are at different ages, but the same person, imaged on day one, imaged one day and then 6 months later again, 6 months later again. Just remarkably few high quality data yet. It's very hard to do. You need a lot of, the imaging has to be done very, very carefully. It takes a lot of time and money. In imaging, time is money, unfortunately. An hour in the scanner costs $1,000 basically. So there aren't that many data sets yet, but I think we are seeing more of it being done and also more of it being publicly shared. So at least with imaging, we can do it. It's just a matter of time when we can actually get access to the data and look at it.
Perfect. And maybe Priscilla can help us to answer this question as well.
Yes, that would be great.
Yeah. Let me see another one. Professor Marimelia asks you, is it possible to incorporate a change in gene expression into functional connective calculations?
Yes, in a sense that I think what you are referring to is perhaps a sort of a manipulation that we would probably do in a model organism, like not in people, but in, let's say, in mouse or in Drosophila. And in principle, yes. So you would compare essentially imaging either before the manipulation and then after, right? Or you could have two different strains of animals, perhaps with the genetic manipulation that has been introduced. And in principle, that can be done. You can make any comparison that you can think of, as long as you can measure connectivity before and after, right? Having said that, getting whole brain data in, especially in rodents, is very difficult. Animal imaging is a whole other technique that requires special expertise, special hardware. The animals are usually anesthetized because they won't hold still in the scanner, and there's massive movement artifact as a result. Then you have anesthesia on top of the manipulation. It gets messy very quickly, unfortunately. So we don't have a very good technique yet that allows us to read out any brain at high resolution continuously, right? We just don't have that yet. What we can do is multi-electrode recordings, embedded arrays, perhaps optical imaging in mouse, for instance, with 2-photon microscopy. Some of the collaborators we have done that. But then you have a very narrow field of view. So you can only see a few hundred neurons, maybe. You don't see the whole brain. So it has its disadvantages also. But the answer to the question is yes, in principle, you can do that as long as you can record either time series or anatomy. And what people have done in laboratories abroad, I believe in Canada, for instance, is to try to knock out hub regions with chemical manipulations in mice and in rats to see how that impacts, on the one side, behavior, on the other side, brain dynamics, for instance. So yes, it can be done.
Yeah, perfect. So Karina Abrão, hi Karina. Karina is also a collaborator here, a professor here. So she's asking, how variable is a resting state connectivity among people and in the same person, considering normal aspects of the life? She means hunger, sleepy,
yes.
Could we decode from the resting state some of the basic sensations or drives of someone?
That is a really great question. It's also a really painful question to answer because the answer is yes, unfortunately, resting state it gives us, I was emphasizing in my talk, the consistency of the pattern we see, right? Remember the two pictures side by side from those two papers, totally different people, different imaging centers, and they are almost on top of each other in the way these different systems are mapped out. That suggests a lot of consistency. But remember, those pictures were group averages, so they were averages over hundreds of individuals. And the averages are very consistent, but the individual variations are quite significant. So number one, there is significant variation even without any other manipulation involved of the kind that you mentioned between people. This is meaningful and important because it allows us to relate these individual variations in how the brain is mapped out and organized to things like behavior and cognition or clinical status. That's being done in many laboratories. But you're absolutely right. There's another sort of important thing to keep track of, and that is things like mood, caffeine intake, sleep status, deprivation, exercise regimes, all sorts of things that happen in the course of our life, okay, our lifestyle variables, nutrition, okay, things like that, and they subtly influence. They do have an impact on the pattern that we see in resting state, and we know this in part because a few individuals in the world have been imaged many times. There's one case of a professor in the US, Russ Poldrack, who spent a year and a half actually imaging himself, I believe, over 80 times. And he kept a very careful record of what he did every day, how he felt, his sleep status, his nutrition, his exercise regime, his mood. He didn't travel very much during this time. He was just going in and out of the scanner, basically. And that data is publicly available. And we and others have looked at it because there's so many different time points in this person's life and so much information about what happened, you know, in the interim, like how did he sleep well, did he not sleep well, did he have coffee, what's the imaging in the morning or in the evening. That's given us a lot of information to answer exactly that question. And the answer is yes, there is impact of things we do every day on how our brain is operating. I remember a picture in a paper, maybe it was just a talk, I forgot, but it showed differences in resting state that were sensitive to caffeine intake. Okay. I know most people on this call probably really need their coffee in the morning, right? And it makes a difference if you go into the imaging center before you had your coffee or after you had your coffee, okay? and so caffeine is a drug, right? And it interacts with brain activity in a complex way, and we see the impact of that even in brain connectivity in a resting state. So this is great in some ways because it's so sensitive to these things. This is fantastic, right? We can do many experimental manipulations, but it's also really problematic because a lot of these factors we can't control, right? And yet they somehow bleed into our studies. So we're interested in finding a biomarker for dementia, let's say, or for autism or what have you. But then there's all these other things going on with caffeine and sleepiness and mood and genetic background and whatnot, and they sort of interfere with the signals we're interested in. This is a problem.
Yeah. Yeah, after all, it's always a problem, statistical problem. No, we always have to have several trials to have a stable average.
Yeah.
In order to have
but the problem is that in a lot of studies, I mentioned earlier, it's really true. I mean, imaging is expensive. So many studies, when they are conducted, really try to minimize the time that people have in the scanner. Because an hour is, $800 to $1,000 depending on where you are, and so also clinical studies. I've been working with a lot of clinical groups over the years, working very closely currently with our imaging center in Indianapolis, where they're studying Alzheimer's disease. people with severe dementia, or even less severe dementia, let's say mild cognitive impairment, really have a hard time being in a scanner. You know, it's not a good environment to be in, and you can't put them in a scanner for several hours in a row. But you would need to do that to get good data. So we always get data that's kind of very shallow, you know, or usually relatively low quality, actually, in clinical studies, because all we can afford, all we can do is 15 minutes, 20 minutes, and it's not enough. So it's a real problem. The data quality increases a lot the more time you spend measuring. Your resolution is improved. You get a better image of what is happening over time. But you can't do it in the people that you're most interested in, the people with conditions, because they really cannot tolerate being in an MR scanner for that long a period of time. And so this is something that we're struggling with in the field in general, I think. And you know, until we have a much more portable technique to read out brain connectivity, we will probably be struggling with this problem for a long time.
Yeah. So let me read another one. Danielle Leal is asking you, well, first of all, he's saying, thank you, Professor Sporns, for your presentation. Very interesting, the perspective of connectivity through the edges instead of the nodes. So do you think this type of analysis would be more accurate to represent the functional connectivity of a given region?
Yeah, it's something that to be honest with you, we don't know the answer to that yet. We have been for the past year really pushing on this very, very hard, especially Rick Wetzel's lab, my colleague here, and Josh, my grad student, and a few other students and postdocs here. And for now, we are focused on laying out sort of, you know, the basic methodology, the basic framework, what are edges, how do we measure that, how do we cluster them, the events I mentioned, which are really interesting. But we don't know yet for sure whether it will be I have to be honest, I mean, we don't know yet whether it will really offer us a totally new insight down the road. We think, yes, we have science indications that this is so, but there's also the problem that when you go to edges, there are a lot more edges than nodes, right? Let's say you have 200 nodes in the brain. That's a decent number of brain regions, 200. That gives you 19,900 edges, n squared minus n over 2, okay, unique edges, that's it. And that means you are now operating on a almost 20,000 by 20,000 matrix. If you go to 400 nodes, which is a better resolution of nodes, then and I've forgotten the numbers, but it's crazy. So unfortunately, you also explode the dimensionality of the problem, right? You have a lot more things to look at now because you're working on edges, not nodes. This can be great if there's additional information, but it can also be bad because you now need to, first of all, your computations take a lot longer, but also you have added potential for overfitting, for looking at noise, basically. So I think that the jury is out. We were able to get some interest in the field for it at this point. Others are already working in this area also, and we're expecting that will increase. But I'm very excited about especially the new type of dynamics that we see, these events, right? This is to me really, I'm working on this myself at home. I mean, I got lots of data running at home all the time, trying to figure out what's going on. Every few minutes, there's this massive burst of co-fluctuation, and it has a common signature. It has, it has that pattern to it. What is that pattern? Why is it there? Why is it between these particular systems that are disconnecting from each other in the brain? We call them events, but are they really events, or are they basically boundaries between what's really important? Let's say trains of thought or mental processes. I think of them sometimes like a screen refresh, right? You basically you wipe the memory of the system. Maybe that's what it is. Okay, so we're kind of hot in pursuit of that, and I'm personally very interested in that aspect of the story. That was disclosed to us when we looked at edges for the first time. So in some sense, that's already given us a new insight, but we don't know yet what it means. And we're pretty sure it's not an artifact, but we have ruled out all the things that we need to rule out, but we can't exclude the possibility that it's something in the end not that interesting. Right now it looks interesting. We'll see where it leads. We have to see where the story leads us.
It looks promising. And by curiosity, and how sensitive it is according to the metrics that you use? I mean, I think you are evaluating only Pearson correlations, but if you use a different type of metrics, I think it will change a lot.
Yeah, the Pearson correlation, it's it's such an embarrassment, isn't it? But with fMRI, the problem is even in a long imaging session, 15 minutes, I showed you the 900 seconds, right? 15 minutes. For the Human Connectome project, that was what the data was from, people were actually in the scanner 4.5 hours, and a lot of data was acquired, including a full hour of resting state, 4 times 15 minutes. That's very rare. Even with the 15 minutes, we only get around 1,100 frames, 1,100 time steps, okay, 1,100 time steps. This does not allow you to do things like transfer entropy. I mean, you cannot compute information theoretic quantities in continuous data to make things even more complicated, because it's not a point process, right? It's a waveform. To get more complex, more interesting measures of dependence, transfer entropy or mutual information or directed coherence or whatever, there's many tools out there. To work in fMRI is almost impossible. And so we're stuck with that embarrassment of the Pearson correlation. I call it an embarrassment because it's literally, it's just so simple, right? And
yeah, but it's hard.
All it does is it's linear dependence. It's a linear estimate of dependency between 2 time courses. That's all. But coming back to your question, of course we can do other recording techniques, right? We could be doing EEG or MEG. We could be doing electrocardiography or ECOG, and record at much higher temporal resolution, right? With EEG, MEG, or ECOG, we can go to milliseconds. And then we get a much longer time series, much more densely sampled data. And in principle, at least, these other techniques then are available to us. However, with EEG and MEG, the issue of localization is problematic, right? Because you're recording outside of the brain, and you have to kind of do an inverse mapping of where signals come from. This is tricky business. It's like a black art. It's like alchemy. And with ECOG, you typically only have limited coverage because the arrays are implanted in patients. And for clinical reasons, there's no experimental leeway, right? You have to kind of go with what the neurosurgeon needs to know. So you only have a certain part of the brain covered and not very densely. So, those techniques have drawbacks also, but at least we can go with much better, more sophisticated techniques, not just cross correlation. Nobody that I know would use cross correlation in EEG, for instance. That would make no sense.
So, okay, so time is flying. Let me read another one here. So Professor Norberto Garcia Carrasco, he's asking, so first of all, he's thanking you. Congrats to Olaf Sporns for the excellent presentation. So his question is related to the need to build tools or algorithms to make the coupling between the brain networks with the expression of their activity to behavior, both normal or pathological.
Yeah, I mean, So that gets to the issue of brain behavior relationships, right? Which is of interest to many people in neuroscience, perhaps most people in neuroscience. We don't just want to observe what the brain does, but we also want to understand how it's driving task-related activity, or how it can account for differences in behavioral performance or cognition. So one of the simplest things that we've done in clinical studies is simply regression analysis. You have some metric that you extract from network analysis, let's say the modularity or the strength of certain connections in individuals. And then for the same individuals, you have some testing in clinical settings that's typically done with standardized tests that probe things like episodic memory or working memory or visual recognition or linguistic capacity and what have you, or general intelligence. And so you have test scores for the individuals that inform you about their performance. And well, the simplest and also perhaps most trivial way of doing comparison would be to have on the one side your brain metric, on the other side your behavioral metric, and carefully accounting for confounds and so forth to simply look at whether they are related to each other, whether something about the brain metric predicts or is associated with behavioral performance or the other way around. More sophisticated approaches go and look at broad domains of behavior and broad domains of brain network activity, and then perform things like canonical correlation analysis, CCA, or machine learning techniques to find mappings from one to the other. That can only be done profitably if you have large numbers of individuals. Otherwise, you tend to fit noise. But we have now databases available, publicly available. Anybody here can have access to data from thousands of individuals. If you're interested in those questions, the nice thing now about data sharing is you can actually do productive, creative science without spending a single dollar of your own money on imaging, right? Because the data is out there. The Human Connectome project involves 1,200 individuals imaging, behavioral cognitive assessment with a battery of tests, task data, anatomy, and even genomics. And it's all free. You just have to go online, sign a simple data use agreement, and then you have access to that data, and you can look at it. This has been another big quantum change really in our field the last decade or so, that these data are now much more openly shared, and this brings into the game people like us, right? I mean, I don't do imaging myself, never have, in part because now the data is out there. We don't have to do this anymore. We have thousands of data sets available, and we can as computational scientists, as people who come from computer science or physics or applied math, we can come in and we can work with this. And this is really something I wanted to stress. I forgot to say it in the talk, that it's really liberating, right? I mean, we don't actually have to do all the measurements ourselves. Spend $30 million as the Connectome project spent to image all these people. We can actually download the data and analyze these relationships and bring new techniques to the table. So I mentioned machine learning, multivariate statistics. There's many other approaches that perhaps we haven't thought of yet.
Yeah, so I think Camilo is already here to finish the section. I'm sorry, the other questions, we cannot read them. So we have tons of questions here. I can send you after this. Maybe we can arrange another seminar.
Definitely. Thank you so much. On behalf of the Graduate Studies and Research Office of the Unifesp, I would like to thank you for this outstanding presentation of today's webinars. And Professor Sponsors, you are more than welcome. We hope that you'll be here at the Unifesp in person. And thank you so much.
I hope so too. I visited Brazil once many years ago now. I really enjoyed my visit, and we have to do it when there's a big soccer tournament. You know, maybe the World Cup or something.
Yeah,
we
definitely.
always have a good.
Yeah, we always have. Yeah, and we hope this pandemic will go away, and then it's very important for us to have you as a collaborator. And thank you so much. And Professor Jean, thank you so much for being in charge of these webinars. And I would like to thank all the participants for being up to this time watching this marvelous presentation, and stay sort of tuned on these webinars. We have different bunches of others upcoming seminars. And thank you so much, Professor Jean and Professor Olaf Sporns.
Thank you.
Thank you,
Goodbye,
Professor.
everybody. Goodbye.
Goodbye. Yeah, thank you very much. Thank you to accept this invitation and to receive Priscilla as well.
Thank you, Jean.
So, thank you.
Bye-bye.
Bye.